everybody, and welcome to Chapter 19. Before we begin, I'd just like to mention that I've been reading through your discussion questions, and I really enjoy reading, what you're thinking, and what you've come up with for all those discussion questions. So keep it coming. So now, and also remember, I'd just like to mention that you should check your email every day and also your Canvas every day as well for announcements and things like that, assignments, announcements, other things. So anyway, let's jump right into Chapter 19. And when we look at Chapter 19, we're going to be talking about viruses. And at first you might think, well, viruses, I don't know if that's as a really interesting topic. But the thing is, though, there really are a lot of really interesting and surprising things about viruses. And most people in general don't know really that much about viruses, not as much as they know about, other parts of biology like cats and dogs or medicinal plants or something like that, But viruses are a really interesting topic that they're learning a lot about currently, even before COVID came along. It's really interesting because so many different things about viruses are just downright bizarre, and it's kind of like you're looking at an alien life form, really. And so here in this first slide, then you see that they've got some drawings of viruses. So here's a virus, and here's another one way in the background, and here's a virus attached to a cell and infecting a cell, and here's a couple more viruses that are farther along getting into that cell. I think this might be HIV virus, I'm not quite sure. But here's another large cell that's being attacked by virus as well. So there's a lot of interesting things and a lot of interesting things about viruses, and study of viruses can be interesting, challenging, exciting, just like any part of biology. So first of all, then, let's look at some kind of basic information about viruses, and then move into a few details about how they operate and how they are able to reproduce and all sorts of things. So a virus, then, as you're probably aware, is very small. It's even smaller than a bacteria, and it's they call it an infectious particle because it's technically not a cell, and we'll get into that in a few minutes. But an infectious particle, it consists of genes that are packaged in a protein coat or covering. So it's really very simple. A really very simple structure, a lot more simple than certainly a eukaryotic cell, and a lot more simple than even a prokaryotic cell as well, a bacteria. So they're a lot simpler than a virus has no way of reproducing on its own. In order to reproduce, it has to actually get into some sort of cell that it infects. And also it doesn't really have any metabolism of its own either. And so it has to rely on a host cell. A host is basically something that's being used by a parasite. And in a way, viruses and bacteria that infect cells are kind of like parasites as well. And so a cell that's being infected by a virus or a bacteria is called a host cell, even though they're an unwilling host usually. But viruses then exist in a shady area between life forms and chemicals. It's too complex than most chemicals, but it doesn't quite rise to the status of a regular life form as Western science would classify it. And it has kind of a borrowed life it's got a borrowed life because it can't reproduce on its own, and it can't do any metabolism on its own. It's got to rely on host cells in order to do either reproduction or metabolism. And here's another, so here's a large human immune cell, a white blood cell human immune cell, and the little orange specks are the HIV viruses then. So a virus then consists of just some sort of nucleic acid, either DNA or RNA, surrounded by a protein coat. So viruses were detected in kind of an indirect way long before they were actually seen. In fact, you have to have a pretty high-powered microscope in order to be able to actually see a virus. And so a little bit of history then. Tobacco mosaic disease, they call it. Tobacco mosaic disease stunts the growth of tobacco and gives their leaves a mosaic coloration. In other words, kind of a spotty, mottled coloration. And in the late 1800s, researchers thought, well, maybe unusually small bacteria might be responsible for it, because they couldn't, when they took samples from these tobacco plants, the infected tobacco plants, they couldn't really see any pathogenic agent, any small protist or bacteria that was causing the disease. So they thought maybe it was unusually small bacteria that were causing it. And they were along the right trail, along the right lines there. And then later, they did experiments, and they saw that the infectious agent that was causing the disease didn't share features with bacteria, such as ability to grow in a nutrient medium. And then in 1935, then Wendell Stanley actually crystallized these, isolated them and crystallized them, and crystallized what we now know as the tobacco mosaic virus, or the TMV. And so he was the first one to actually isolate a virus then, 1935. So they extracted sap from a tobacco plant that had the mosaic disease. They passed it through a filter that's known to trap bacteria, and they rubbed the filtered sap then on healthy plants, and the healthy plants became infected. So basically, they took material from an infected plant, they filtered out all the possible bacteria, and with the liquid then that was the result, it was still able to infect healthy plants. So they knew that it was something that was smaller than a standard bacteria then. So viruses are not cells, because they can't reproduce on their own, they can't carry out their own metabolism, et cetera, et cetera. A virus is a very small infectious particle. It has nucleic acid, like we said, enclosed in a protein coat, like we said. But in some cases, though, there's a layer outside the protein coat called a membranous envelope. So a viral genome, then, all of the genes, the sum total of all of the genes that they have, viral genomes consist of either double or single-stranded DNA or double or single-stranded RNA. So here's kind of a specialized case in which RNA can actually be double-stranded. So viruses are classified as either DNA viruses or RNA viruses. Pretty straightforward, pretty convenient. And it's either a single linear nucleic acid molecule, or it can be a circular nucleic acid molecule. And viruses can have anywhere between 3 and 2,000 genes in their genome. The virus with just 3 genes, that's a pretty small, pretty stripped down version of very straightforward, stripped down version of a virus. and so up to maybe 2,000 genes in their genome. And compare that to like humans that have a little over 20,000 genes in their genome. So the large complex viruses can be pretty formidable in terms of numbers of genes in their genome. A capsid, what's a capsid then? It's a protein shell that encloses the viral genome. And capsids are built from protein subunits called capsomeres, because mere means part, like a polymer. A polymer is something that's in many parts, like a nucleic acid is a polymer, because it's made of many different nitrogenous bases, one right after the other. So a capsid then can have a variety of structures and shapes, and viruses then could be referred to as either helical or icosahedral viruses, depending on the shape of the capsid. So here's a helical capsid, and they're pointing to just one capsomer of the capsid. So this capsid basically is a protein coat that's arranged from proteins that are in a very helical spiral sort of shape. Then this is icosahedral, and three-dimensional geometric figure, basically. And you can see there, here's an RNA virus, so you've got RNA inside it, and protein surrounding the RNA, and they've got a polymerase as well that can produce more RNA, and then you've got a membranous envelope then, and glycoproteins, spikes. Here's another one that looks like some sort of alien spacecraft or like a lunar lander or something, that's got this apparatus here to be able to land on and attach to host cells. And here's an actual photograph of it in an electron microscope. In fact, here's actual photographs of all of these things under electron microscope. So really interesting. So viruses come in kind of a wide variety of shapes then, shapes and sizes and things. So some might have accessory structures to help them infect hosts. For example, here you've got accessory structures to help them infect the host cell. So viral envelopes derived from membranes of the host cells. Derived from means modified from. They take parts of the host cell membrane and they modify it to make a viral envelope. These viral envelopes then can surround the capsids of influenza viruses and other viruses as well in animals. Viral envelopes can contain a combination of viral envelopes can contain a combination of viral and host cell molecules that are modified according to what the virus needs, and things called bacteriophages, or just referred to as phages, then are viruses that infect bacteria. So it's got to be a pretty small virus to be able to infect a bacterial cell. And they have an elongated capsid head that encloses their DNA, and then they have a protein tail piece that attaches attaches this phage or bacteriophage to the host cell and injects the DNA inside. So this right here is a bacteriophage then. So it's got these accessory structures then to be able to attach to the bacteria cell and inject the DNA inside the bacterial cell. So then viruses are obligate intercellular parasites. Something it's an obligate parasite. In other words, it's obliged to be a parasite. It's required to be a parasite. It can't live any other way. So it's an obligate intracellular. So inside the cell, intracellular. So they can only replicate or reproduce inside the host cell. And each virus then has a host range, a limited number of types of host cells that it can infect. Because each virus cannot infect just any cell from just any species. There's only a limited number of types of cells that they can infect. The viral genome enters the host cell in a variety of ways, and we'll see those throughout the chapter here. But once the viral genome has entered a cell, the cell is forced to manufacture viral proteins, and also viral nucleic acids as well. And so the virus uses host enzymes and ribosomes and tRNAs and amino acids and ATP and other molecules that it needs in the host cell in order to manufacture viral proteins and viral nucleic acids. And so then these viral nucleic acids and the capsomeres spontaneously self-assemble into new viruses, then new virus particles. And so they've got a nice diagram of the whole process. So here's a virus, and it's got DNA inside, and it's got a capsid on the outside. And so the first step is that it enters. It enters the host cell, and the uncoating, which means the capsid is removed so that the DNA can come out and freely start working. So then the viral DNA goes through replication. So there's a lot of viral DNA, and the viral DNA affects the mRNA of the cell, and it causes transcription then and manufacture of capsid proteins. And then all of the viral DNA and viral capsid proteins are assembled together then into new virus particles. And these virus particles can exit the cell and infect other cells, other host cells. And here's an animation you can watch about viral replication. So the phages then, these phages are the best understood and the most intensely studied of all viruses. And they have two alternative or alternate reproductive mechanisms. They go through a lytic cycle, or they can go through a lysogenic cycle. And so the lytic cycle or lytic cycle, lytic actually means to break something down. Like if you're analytic, you're able to break down things and explain them better and understand them. Catalytic is to break things down, like catalysis. Catalysis is chemical processes and chemical reactions that break down molecules into smaller pieces. So lytic actually means breaking down or breaking apart. So the lytic cycle then is a replicative cycle that basically causes the death of the host cell. And we'll see a diagram of this. And so basically in the lytic cycle, it produces new phages, and it lyses or breaks open the host cell wall or cell membrane in order to release the viruses, the progeny viruses that have been produced. And a phage that reproduces only by the lytic cycle is called a virulent phage, because it causes a very serious disease then. And so we can look at this step by step. So a phage then attaches to the outside of a cell, and then the phage DNA basically enters the host cell, and the host DNA is degraded, and then the DNA of the phage causes the synthesis of viral genome and viral proteins. And then those viral genomes and proteins are assembled into new virus particles, complete new virus particles. And then the cell bursts open to release the new virus particles into the atmosphere, or into the environment then. So the head is assembled like this, the tail is assembled in pieces in steps like this, and the tail fibers are first assembled as different parts and then put together and assembled into the whole phage assembly. And they don't know exactly how it's assembled. There's probably special enzymes that do this, and probably further study is going to actually find out exactly how these new phage parts are assembled together. And so basically then, here's the release cycle when the new virus particles are released. So this looks like some sort of alien movie or something going on. And then so here's an animation to show the lytic cycle in a phage life cycle. So that's the basically that's the lytic cycle, and now we're going to go on to the lysogenic cycle. What happens there then? This lysogenic cycle replicates the phage genome without destroying the host. So the viral DNA then, or the viral genome, is incorporated into the host cell's chromosome. And so basically it becomes part of the host cell's chromosome, and whenever the host cell's chromosome is replicated, it'll also replicate the phage genome. And phages that use both the lytic and lysogenic cycles are called temperate phages. And so basically, a phage can do a lytic cycle and a lysogenic cycle. And so basically in the lytic cycle, as soon as new bacteriophage DNA is produced, it can either decide to do a lytic cycle from there, or it can decide to do a lysogenic cycle from there. So in other words, produce new virus particles, or just have the phage virus DNA to be incorporated into the DNA of the host cell. And when it's incorporated into the DNA of the host cell, it's called a prophage. And then when the host cell itself divides, then each of those new cells then will be infected by the virus prophage. So it's kind of an interesting life and an interesting process. And here's an animation then that you can watch on your own time about the phage, lysogenic, and lytic cycles. So the viral DNA that's integrated into the host DNA is known as prophage, like we saw on the diagram. And then every time the host cell divides, it copies the phage DNA along with its own chromosome and passes it on to the daughter cells that are produced. And some sort of environmental signal can trigger the virus genome to actually exit the bacterial chromosome and switch to the lytic cycle, or the lytic mode. But bacteria are not left without defenses against phages. So natural selection actually favors bacterial mutants that have surface proteins that cannot be recognized as receptors by a particular type of phage. So in order for a type of phage to actually find the right host cell to infect, the phage has to find surface proteins on the bacterial cell that can serve as receptors for this phage. So if bacteria will actually mutate or change somehow genetically. They can actually change maybe their surface proteins so that they're not recognized by the phage, and therefore the phage doesn't infect them. Foreign DNA prophages can be identified as foreign and maybe cut up by cellular enzymes known as restriction enzymes. The bacteria host cell restriction enzymes. So the bacterium, and bacterium is just singular for bacteria. So bacteria is actually plural. Bacteria is actually plural, but bacterium then is the singular of that. So bacterium's own DNA is protected from restriction enzymes by being methylated, having a lot of methyl groups on it. So that's how a bacterium's own DNA is not cut by its own restriction enzymes. So both bacteria and archaea, which are also prokaryotes, remember, both bacteria and archaea can protect themselves from viral infection with something called a CRISPR-Cas9 system, or a CRISPR-Cas system. And these are sequences that are called CRISPR, that is basically clustered regulatory interspaced short palindromic repeats, because they're called palindromic because their DNA sequences are actually palindromes. So these are CRISPRs for short. And so let's explain what these actually are then. So each spacer sequence between the repeats corresponds to DNA from a phage that had infected the cell before. Particular nuclease proteins on the host cell interact with the CRISPR region, and these are called CRISPR-associated proteins then. When a phage infects a bacterial cell that has the CRISPR-Cas system, the phage DNA is integrated between two repeat sequences. And if the cell survives the infection, it can block any attempt of the same type of virus to reinfect it. The attempt of the phage to infect the cell, to reinfect the cell actually, triggers the transcription of the CRISPR region, and the resulting RNAs that have been transcribed and produced are cut into pieces and bound by Cas proteins. And then the Cas proteins use the phage-related RNA to target invading phage DNA. So it uses the phage-related RNA as a primer in order to recognize invading phage DNA. And then the phage DNA then is cut and destroyed so that the infection doesn't take place. In natural selection, but the thing is though, natural selection is working on the phages as well, and natural selection favors phage mutants that can bind to altered cell surface receptors that are resistant to enzymes. The relationship between then the phage and the bacteria, it's kind of like an arms race. The bacteria make an improvement that helps fight against the phages. Then the phages make an improvement that helps circumvent whatever new strategy the bacteria has. And so it's a constant battle back and forth. It's like an arms race. And so basically then, and here's what we see, here's what we've just discussed then in the last few slides. So infection by a phage virus then triggers transcription, infection, and introducing the DNA from the invading phage. That basically stimulates transcription of the CRISPR region of the bacterium that contains DNA from a previous infection by that type of phage. And that so basically then transcription of the CRISPR region produces an RNA transcript, and after it's processed, the RNA transcript is basically the processed RNA is basically attached to a Cas protein, and the Cas protein then has active sites that cut DNA, and so it recognizes then it uses the RNA that it's produced to recognize the DNA of the phage DNA. And so when the phage DNA is recognized, the Cas protein then cuts the phage DNA so that the phage DNA cannot infect the cell, and it can no longer replicate. No longer has the ability to replicate. And here's just a detailed look in two parts of that diagram we just went through. So and here's a detailed model of basically the Cas protein with the RNA transcript in it. Well, the RNA and the DNA. So it's kind of cool. It's in kind of cool colors. So this is basically the end of part 1 then of the chapter 19 lecture, and stay tuned then in another day or so, and I'll put up part 2 then of chapter 19. And like always, remember that that if you have any questions or would like any sort of explanation, or if you need any help for anything, just let me know, send me an email or whatever, and I can answer your questions, and we can go on from there. So keep studying, keep checking your emails and your announcements, and we will see you again later for another lecture, and it will be part 2 on chapter 19 on viruses. Thank you.